Good afternoon. I'm Jason Karlawish, co-director of the Penn Memory Center and a professor in the School of Medicine at the University of Pennsylvania. Thank you so much for joining the webinar, Are We There Yet with PET, Amyloid PET as a Surrogate Endpoint for Clinically Meaningful Outcomes. I'm here as co-chair, along with my colleague, Dr. Josh Grill, of the Alzheimer's Clinical Trials Consortium Internal Ethics Committee. We welcome you to our webinar. Since June of 2020, 3 treatments have been reviewed by the FDA using the accelerated approval mechanism. 2 were approved and 1 turned down. The committee was impressed that science proceeds by open and collegial debate and discussion, and has felt that that is needed for this topic. Therefore, we organized this debate. Just a moment. I will now switch the slides. Our first presenter, will be Holly Fernandez Lynch. She's an assistant professor of medical ethics and law at the University of Pennsylvania. Her work focuses on clinical research ethics and regulation, access to investigational medicines, outside clinical trials, and FDA pharmaceutical policy. She declares that she's funded by the Arnold Ventures for work related to FDA approval standards and post-market studies. She will be speaking on background and overview around the law and regulation for surrogate endpoints. Following Dr. Fernandez Lynch will be Dr. Dennis Selkoe. He's the Vincent and Stella Coates Professor of Neurological Diseases at Harvard Medical School, co-director of the Ann Romney Center for Neurologic Diseases at Brigham and Women's Hospital. His research on beta-amyloid protein and amyloid precursor protein led to the amyloid hypothesis of Alzheimer's disease and has focused on translating these discoveries into therapeutic options for Alzheimer's disease. He is the director and provides consulting for Athena Biosciences and has served on his advisory board and consultant to Eisai. Next, Dr. Chris Van Dyck will present on amyloid PET not being a suitable surrogate for endpoint for clinically meaningful outcomes and the accelerated approval of Alzheimer's drugs. He is a professor of psychiatry, neurology, and neuroscience and the director of the Alzheimer's Disease Research Unit at the Yale School of Medicine. His research focuses on the neuroimaging and therapeutics of Alzheimer's disease and brain aging. He declares that he's an advisor or consultant for Roche, Eisai, Ono, and Cerevel Pharmaceuticals. After Dr. Van Dyck presents, Drs. Van Dyck and Selkoe will have 5 minutes to cross-examine each other. And you all then will be presented this question to vote on amyloid PET. Are we there yet with amyloid PET as a suitable surrogate endpoint for clinically meaningful outcomes and the accelerated approval of Alzheimer's drugs? Your choices will be to vote yes or to vote no. You can publicly explain your vote as well by emailing Terry Casey at the email address that you see on the screen. We will compile those public comments and post them on the website where the recording of this debate will also be available. And you will be notified about when that all is available. This slide will appear again during the debate. Let me now turn it over to Holly Fernandez Lynch to begin the discussion. Holly?
Great. Thanks so much for having me, Jason. I am going to share my slides here. Can you see that okay? I hope. All right, great. Well, so everyone, I have been asked to kind of lay the groundwork from a legal perspective of what FDA is looking for in order to accept a surrogate endpoint in support of new drug approval, and in particular to accept an endpoint to support an accelerated approval. So let me start just by situating us in the past, century plus a few decades of FDA's history. So starting, in the early 1900s, FDA was on a course of requiring more information about drugs before they would be allowed on the market. First making sure that they were not mislabeled or adulterated, then making sure that they FDA met a standard for safety, and then in 1962, adding a requirement that they be demonstrated not only safe, but effective before they could be marketed for sale, right? So we see over the course of the 20th century, this trajectory towards increasing certainty about the quality of drugs allowed on the market. But these requirements, of course, decreased the speed with which products were allowed to enter the market. And so that went on, basically until the early 90s in response to pushback from the HIV/AIDS advocacy community, right? So we see in the midst of this epidemic crisis, advocates pushing back against FDA, saying, look, we have a deadly disease. There are no good treatment options for us. We want you to allow for some flexibility, accept some uncertainty, allow us to make our own decisions about which products are promising enough for us to take them. We have no time to wait. And what the HIV advocates succeeded in doing is getting FDA to change its mindset a bit. So over, most of the 20th century, FDA had been protecting patients by keeping unsafe and ineffective products off the market, and that is still its statutory mandate. But now it has progressed to also recognize that it needs to play a role in facilitating access expeditiously to promising drugs. So one outcome of this HIV advocacy was the adoption of the accelerated approval pathway in 1992. So what we see today is really a reversal of the trajectory that FDA had been on. We see increasing attention to speed, right? We need to get products to patients with unmet treatment needs as quickly as possible, right? This is the mindset. And we might be willing to accept greater uncertainty about those products. There's in the text box at the top here, this is a quote from some recent FDA draft guidance that explicitly acknowledges that it might sometimes be more willing to accept false positive conclusions, right? Less certainty about effectiveness when you're thinking about the possibility of rejecting, right? Failing to approve a potentially effective therapy. So the standard for FDA to approve a new drug is set by law in the Food, Drug, and Cosmetics Act. This particular statutory standard has been in place since the early 1960s. And what's required is that drug sponsors provide FDA with full reports of investigations to show that the drug that they're seeking approval for is safe and effective for use. So FDA, when it's evaluating safety, will reject a drug if the sponsor has provided inadequate information to determine that the drug is safe, right? There's too much uncertainty about that, or if the results show the drug is in fact unsafe. In terms of effectiveness, what the statutory standard calls for is substantial evidence. So in the absence of substantial evidence that the drug will have its purported effect under the labeled conditions, FDA is directed to reject, to refuse approval for those drugs. Now, it's not just any purported effect. That effect also has to be determined by FDA to be clinically meaningful. So that substantial evidence standard gets further defined in the relevant statute to be evidence consisting of adequate and well-controlled investigations on the basis of which it could be fairly concluded by experts that the drug will have the purported effect. So that adequate and well-controlled investigations, right? That's what we're talking about when we think about substantial evidence of effectiveness. And that's a reference both to the quality of the evidence, right? It has to be adequate, it has to be well-controlled, and to the quantity of the evidence. So you can see I've highlighted the S in investigations. The traditional expectation was that you need at least 2 adequate and well-controlled investigations to support FDA approval, but that standard has weakened over time, and FDA is often willing to accept only 1 pivotal study in support of drug approval. Now, how do you show that you have substantial evidence? This is where accelerated approval comes into play, right? So first, you can show substantial evidence by demonstrating an effect on clinical endpoints, right? Clinical endpoints are direct measures of how the patient feels, functions, or survives right? Things that they can feel themselves. No, you're not relying on a laboratory test or, a radiographic image or something along those lines. This is something that is a direct measure, and that will support traditional approval. The other way that you can get traditional approval is showing an effect on validated surrogate endpoints, right? So a surrogate is an endpoint that is not a direct measure of clinical benefit, but validated surrogates have proven themselves to accurately predict clinical benefit, right? So clinical endpoints, validated surrogates can support substantial evidence in favor of traditional approval. The accelerated approval standard allows a different type of endpoint to be used to meet the substantial evidence standard. So first, we can rely on unvalidated surrogate endpoints. These are surrogates that are deemed reasonably likely by FDA to predict clinical benefit. And then we might also rely on intermediate clinical endpoints, right? So these are not surrogates, they are direct measures of clinical benefit, but it's something short of irreversible morbidity or mortality, right? So if you show an impact on an acceptable unvalidated surrogate or an intermediate clinical endpoint, then FDA will grant accelerated approval. And the idea behind acceptance of these weaker endpoints is that they might be able to be tested and evaluated more rapidly than the traditional clinical endpoints or validated surrogates. Now, FDA is adamant that the standard for accelerated approval is not a weaker standard. The reason they say that is because it still has to meet the substantial evidence standard. Nonetheless, it is clearly weaker evidence in support of approval, right? If you are relying on an unvalidated surrogate endpoint, there is, of course, uncertainty associated with that. Because it is a weaker standard of evidence, the accelerated approval pathway can only be used in certain circumstances, right? You have to have a drug that is intended for use in a serious or life-threatening disease. And very importantly, if you get your drug approved through the accelerated approval pathway, FDA has authority to require post-approval trials to verify that predicted clinical benefit, right? So the sponsor will be required by law to conduct these confirmatory studies. And if those confirmatory trials fail to verify benefit, or if the studies are not done in a timely fashion, then FDA has an expedited withdrawal pathway that is not particularly expedited in practice, but by law it is supposed to be expedited. So these are the safeguards that are associated with accelerated approval, which is intended to be a compromise. We get promising drugs to patients more quickly, and then we confirm their benefit later. We know, and FDA often is on the record as saying that this is not going to be a perfect process. There are going to be some drugs that get accelerated approval that don't pan out, right? We know that this is a trade-off. And so the name of the game is to figure out how we can minimize the risks associated with this pathway. One way of doing that is minimizing bad guesses by selecting reasonably good surrogates to rely on for support of accelerated approval. We also need to make sure we have speedy confirmatory trials and rapid withdrawals when things don't pan out. So let's talk a little bit more about how FDA thinks about surrogate endpoints, right? So again, these are just markers, not measures of direct clinical impact, right? But a laboratory measurement, a radiographic image, something that is thought to predict clinical benefit. And a surrogate will go through this kind of trajectory where it starts out just as a candidate, couldn't be used to support any kind of FDA approval when it's still just kind of being examined for whether it might be valuable in predicting clinical benefit. Once it moves beyond just being a candidate surrogate endpoint, FDA might decide, we think this is strong enough that it meets that statutory standard of reasonably likely to predict clinical benefit. FDA decides this could support accelerated approval. And then once the endpoint gets validated, right, then we know it predicts clinical benefit and it could support traditional approval. Reasonably likely to predict clinical benefit is a very fuzzy standard, right? It requires a lot of discretion and judgment. So FDA has issued guidance about what counts as reasonably likely to predict clinical benefit. There's a little bit of guidance in the statutory language itself, right? It says, what's the type of evidence you can look at to determine whether a surrogate endpoint is in fact reasonably likely to predict clinical benefit. And the statute says you can look at epidemiological, pathophysiological, therapeutic, pharmacologic, or other evidence, or other scientific methods or tools, right? So they're basically saying you could look anywhere to gather support to justify the notion that a particular unvalidated surrogate might be reasonably likely to predict clinical benefit. The guidance goes on to emphasize the importance of considering the biologic plausibility of the relationship between the endpoint of interest and the disease. And it emphasizes that just showing pharmacologic activity of the drug on its own is insufficient, right? It needs to be on some endpoint that you think might actually be meaningful down the road. A little bit more guidance is offered where FDA indicates that there's a couple of tricky things about specifying an appropriate surrogate endpoint, and addresses areas where it might be quite difficult to have confidence in a surrogate. So one challenge is, what do you do when you don't have a strong understanding of the disease process, right? If you're not quite clear on what the underlying cause of the disease is, or what the disease pathway is that's ultimately resulting in clinical harm, then it becomes very challenging to know whether an effect on a surrogate is likely, to represent a meaningful effect on this causal pathway that's resulting in clinical harm, right? And similarly, the more you understand about causation of the disease, the causal pathway, how the endpoint intersects with that causal pathway, the greater confidence you can have. And so then FDA says, look, to the extent that you understand the connection between the abnormality of interest, right? Whether it's, accumulation of amyloid plaques, or, the level of neurofilament light chain, protein in a patient's blood, right? These are things, the more you understand the relationship between those abnormalities and the clinical outcome, the stronger basis you have for concluding that an effect on that endpoint would lead to an effect on the outcome of interest. However, right, despite a couple of pages of guidance about this, FDA recognizes that all of these judgments are case specific and not readily generalizable. And they basically say, we're going to look high and low. We're going to look at all the evidence. We're going to consult our external experts. And recently, the FDA Office of New Drugs Director, Peter Stein, gave a presentation at a National Academy of Medicine meeting focused on accelerated approval. His whole talk was about how FDA evaluates possible surrogate endpoints. And here are a couple of choice quotes from his presentation, right? We don't have a simple formula or algorithm. It requires a deep dive into the data supporting the surrogate. This is always a challenge. And then he articulated that they really have to evaluate how risky, are they willing to be, right? What is the risk of error that FDA is willing to accept? And acknowledging, right, that it is not going to be the case that every accelerated approval surrogate is going to pan out. And in fact, he argued that if they did, then FDA would be too conservative, right? They wouldn't be using the accelerated approval pathway as it was intended. So very briefly, a couple of critiques of the current approach that FDA takes to evaluating potential surrogates is that there's no clear standard for determining what magnitude of change in a surrogate is likely to be clinically meaningful. That's obviously something that's relevant in the context of Alzheimer's disease and the drugs that have recently been granted accelerated approval. And then there's a lot of concerns about the procedural challenges, right? So if we don't have a cut and dried standard that we can apply, then you might say, well, at least FDA should be transparent about their judgments and the principles that they're using. But there's no requirement to consult an advisory committee when considering acceptance of a new surrogate or allowing the public to comment. And as we've seen, FDA might accept a surrogate even when clinical endpoints were evaluated and not met. And there's also criticism about what's referred to as the pipeline impact of accepting a new surrogate. So once FDA grants an accelerated approval on the basis of a particular new unvalidated surrogate, other drugs targeting the same mechanism of action can also get accelerated approval on that basis, unless and until that surrogate is invalidated. And so it can really shape the clinical pipeline of drugs that are coming on the market and where research funding and attention is going. Now, at the end of 2022, there was some new legislation passed relevant to FDA, and FDA has been directed specifically to develop 2 new guidances to provide a bit more detail on surrogate endpoints over the next 18 months. So I'll close just by, saying that a lot of the recent controversies around accelerated approval have resulted in attention to problems with confirmatory trials, the fact that they're often delayed, they might not be well designed, as well as slow withdrawal on the part of FDA when accelerated approvals don't pan out. But it is also really important to sharpen those criteria for determining which unvalidated surrogates are reasonable to accept as a basis for FDA approval. So let me.
Thank you, Holly.
Thank
you, Dr. Fernandez Lynch. Thank you very much. Next up will be Dr. Dennis Selkoe, arguing for 20 minutes the case for beta-amyloid as measured by PET. You'll have 20 minutes to do this, and then we will turn over to Dr. Chris Van Dyck for the case against beta-amyloid as measured by PET. They'll have 5 minutes for exchange, and then we'll put up the poll to vote on the question. Dr. Selkoe, if you want to, your slides are showing up. You're doing great. Slideshow, there you are. And there you go. And I'm going to unmute your video and audio. Your slides are visible. Just need to unmute your video and audio. Audio is unmuted. Just your video now. If you want us to see your charming, smiling face.
Right. And let's see where that is.
It's above your neck and below.
Yeah. No, that's right. I want to make sure this is. I don't quite see the button for. Oh, here it is. It's just appeared. Yeah.
Yeah, I know it's funky. Oh.
Okay. Good. How about now? Can you see everything, including myself?
Slides are visible. You're visible, and your audio is good to go.
Good. All right. Well, thank you very much, Jason, for inviting me. And I have a wonderful, very highly respectable adversary, so to speak, not in real life, but I'm delighted that Chris is going to take the other side, and I'm sure we'll come to a rational conclusion. I think a lot of what you just heard from Holly are standards that will be possible to meet with what I'm about to tell you in terms of the reasonableness of amyloid as a surrogate marker for accelerated approval in this time. And Chris added the point suitable. So what I decided to do was to go through 2 lines of evidence broadly, the science behind why amyloid is central and many of us feel causative of the pathology and the clinical deficits of Alzheimer's, and then some of the clinical research that also supports the notion that amyloid links very closely to cognitive function, behavior, et cetera, as we've seen from the recent lecanemab data. So let me begin with this image of the amyloid PET scanning that we're debating here. And I'll tell you that amyloid PET has been an incredibly powerful tool to help confirm the amyloid hypothesis. And we know that amyloid in Alzheimer's disease, its deposition is often followed and commonly followed by buildup of tau that already appears even before amyloid does on PET scans in the medial temporal lobe, but then seems to spill out of that area and spread, as we call it, to other regions of the brain. So it's wonderful to have these powerful imaging methods, and I'm going to give you a series of data that support the notion that amyloid buildup is essential to Alzheimer's and leads on to tau alteration and then to cognitive impairment and dementia. And then I'll show some data from others about how amyloid does predict the high likelihood of cognitive decline over time. So let me just go here. So just briefly, I'll remind you of what we've come to know in the last 30 years. The amyloid precursor protein diagram is a simple stick diagram at the top of a canonical round cell, undergoes progressive cleavage, first by beta secretase to release the large ectodomain, and then by a curious cleavage inside the membrane to release A beta. So this happens in essentially all mammals. It certainly happens in all primates that have been examined. And A beta monomers are not believed to be toxic, but they can dimerize over time, especially with age, and trimerize and form higher aggregates referred to as oligomers. Those in turn can further polymerize into protofilaments and into fibrils, and then into amyloid plaques. And we see an example of a patient whom I followed during life that developed the disease around 69 years old with typical spherical plaques. You see two examples of those with my cursor here, and adjacent neurofibrillary tangles, all of this in the amygdala. And this happens in very large amounts, largely in areas important for behavior and cognitive function. Now, the plaques are associated with profound microglial and astrocytic changes, and this has been known for decades. And so the microgliosis and astrocytosis focuses on the amyloid plaques and is a sign of local inflammation. It's very unlikely that having these in the brain is in any way neutral or beneficial. And so I will be making the analogy to other chronic diseases in this discussion, where we have found surrogate markers that predict the likelihood of decline and therefore are worthy therapeutic targets. And I very much believe that amyloid buildup is one of those. The hypothesis was that Alzheimer's would turn out to be a syndrome caused by an imbalance between the production and clearance of the amyloid beta, especially the A beta 42 residue form, that leads to its accumulation and aggregation in brain regions serving memory and cognition. And this imbalance could be caused, we thought in early days, by numerous different genetic and potentially environmental factors. And familial Alzheimer's disease provided the linchpin for understanding that indeed amyloid buildup could be causative, at least in some humans. Deciphering the genotype-phenotype relationships in familial AD strongly supports this notion that A beta buildup is not a good thing and needs to be removed if possible. And here's a summary of the first four major genes that were described. And in each case, these mutations, or in the case of APOE4, a polymorphism, has the arrows pointing up. In my lab and Steve Youngkin's lab, we figured out these relationships in simple cultured cells. And in each case, A beta buildup or lack of disposal leads to increased density of plaques and increased production, in some of these cases, all lifelong. And at the bottom of the cell, you can see that a number of other genes, there are now upwards of 70 genetic risk factors that have been identified, and about a third of those directly affect the amyloid cascade, the conversion of APP to amyloid. So lots of strong genetic evidence. This is perhaps the most striking, the situation in Down syndrome, a patient whose brain I obtained at Children's Hospital when the young man was 12 years old when he died. And by that age, he had a cerebral cortex full of diffuse A beta deposits that lacked microgliosis, astrocytosis, neuritic dystrophy. So the science tells us that in Down syndrome, which is a type of a phenocopy of Alzheimer's disease and is indistinguishable under the microscope unless you know the patient's chromosomal status and age, Down syndrome leads to first A beta deposits that lack other cellular features, and then those features develop over time, and these patients eventually lose further ground cognitively. And one of the things we learned was that the enzyme that creates amyloid beta, this is a diagram that shows that, is a 9-transmembrane complex called presenilin. Some years ago, we found that it was the catalytic core and used 2 aspartate residues in transmembrane 6 and 7 to cleave APP, this is the APP molecule, in the membrane and release the amyloid beta peptide. So presenilin, which is by far the most common site for mutations that cause admittedly rare familial forms of Alzheimer's is the actual enzyme that produces. So there's great symmetry in Alzheimer's genetics. All of the dominant mutations that cause early onset disease are either the substrate, APP, or the enzyme, presenilin, that creates the amyloid beta peptide throughout life. Now, the striking finding in the Icelandic mutation by Carrie Stephenson and colleagues that there can be mutations in APP, the precursor, that decrease amyloid production lifelong. And that's an example here with about a 40% decrease in A beta production by cells that express this so-called Icelandic mutation, which occurs at the second amino acid of the A beta region. And this offers lifelong protection. And it was shown even in autopsy studies of a Finnish patient who carries this protective mutation, that there was merely a wisp of amyloid at age 104, and the patient was cognitively intact until quite close to that age of death. And now we can go on and demonstrate mechanistically that this genetic evidence that A beta could even be causative of the disease, or maybe in many cases, can be associated with the other phenomena of Alzheimer's disease. Here we isolate amyloid monomers, A beta monomers and A beta dimers, by size exclusion chromatography and show them on a Western blot compared to control extracts of a normal brain. And the AD brain extract has abundant monomers and dimers and higher oligomers that don't stay intact in this denaturing gel. And those fractions, like fraction 8 and 9 that are dimer rich, fraction 9, 10, and 11 that are monomer rich, can be floated onto, that is applied to, healthy neurons, rat neurons or human neurons. Both have been done. And you can see here that the control brain fractions that are analogous to the ones in Alzheimer's do nothing to the beautiful neuritic morphology of these neurons. Neither does the A beta monomer rich fraction from an Alzheimer brain, serving as an excellent control, but the A beta dimer rich fraction causes beaded neurites, dystrophic neurites that we would liken to the abnormal neurites that are around essentially all neuritic plaques. Now, if you look at tau, you see elevated phospho-tau, one of the hallmarks of the tau abnormality in Alzheimer's. So AT8 is a epitope that's long been used to study human Alzheimer tangles, and it is elevated by A beta oligomers harvested from our patients' brains, from typical Alzheimer brains, and applied, but not if the A beta oligomers are first depleted with an antibody. So the phospho-tau abnormality that's so much studied in Alzheimer's is beautifully induced by the application of soluble A beta oligomers from bona fide Alzheimer patients from postmortem brains. And curiously, interestingly, if you delete the tau through genetic engineering, through RNAi approaches, now tau, of course, is gone in these two panels because it's been knocked down, but you see the A beta oligomers no longer change the neuritic form in these cultured neurons. So you need tau, and tau is permissive and supports the clinical evidence that A beta buildup leads to more and more tau abnormality that can be seen with biomarkers and with imaging. This kind of system can be used to discover antibodies, such as the one that our field is very focused on. So, for example, this is without antibody treatment. As I showed you, these neurites become degenerative and neuritically dystrophic. But if you add, just float onto the neurons, 3D6 and A beta N-terminal antibody, 8AT1, another one, you can rescue this effect. So you can show immunotherapy in a dish long before we actually had clinical evidence that these could do something about clinical progression. But C-terminal antibodies didn't do this. So a lot of basic science evidence that it's not just A beta is a problem, but it leads to tau alteration. And if you didn't have tau, I wonder whether you would get the same degree of Alzheimer's. Probably it would take longer or not be as profound. We've gone on to actually take oligomer selective antibodies and show that they can rescue long-term potentiation, a classical correlate of learning and memory in slices of mouse brain. So here in red is an A beta oligomer preparation from a patient's brain applied and interrupting LTP, and that's rescued in blue with the presence of an antibody. And we then use that antibody to actually immunopurify the oligomers and show that they can be applied to neurons, healthy neurons, and directly inhibit long-term potentiation, which so many of us in basic neuroscience feel is a valid correlate of decline in memory and learning. We've developed an assay that quantifies these A-beta oligomers from human brain and can see them in CSF and in plasma. Here in CSF from our own clinic, the amount of that A-beta oligomer amyloid in CSF correlates with the well-known ADMAR commercial assay to measure total tau and phospho-tau at PT 181 position. So an important correlate with a diagnostic measure we use in the clinic essentially every day. What are your CSF tau and phospho-tau levels? They are predicted by A-beta oligomer in that same CSF sample. And we can then now move from some of the basic science that supports the centrality of amyloid buildup as a strong negative for function to what has been seen, for example, in Chris Rowe's study here, about the prevalence of Alzheimer's disease over time during the aging process in the last few decades of life, and how that relates to the prevalence of plaques by immunohistochemistry in the brains of patients, of multiple patients. And so what Chris Rowe pointed out is that PIB-positive PET lies right nicely along the counting of plaques by IHC and postmortem tissue samples. So PIB positivity is, as we all now believe, reflecting what happens in terms of plaque deposition, giving us 15 years or so before the prevalence of AD rises. And that's an interval between A-beta deposits in the brain and cognitive decline that I think the FDA needs to use all of the tools available to allow therapies to come to our patients during those 15 years and to wait as little as possible. So if we think of moving towards disease-modifying treatments and prevention of Alzheimer's disease, one of the big problems we face in this debate about whether amyloid is a surrogate marker of outcome clinically is that there's so much common knowledge among everyone walking up and down the street that amyloid lowering doesn't work. And this slide is very important to dispel that. I won't go through it in the interest of time, but there are a number of trials, 10 of them listed here, and there are others in the literature that I've looked at closely, and their target and mechanism was to do something about A-beta levels, A-beta oligomers, moving A-beta monomers in the case of solanezumab. All of them had problems with the trial conduct and with how they developed agents that would help, and none of them actually lowered amyloid. So the notion that you hear in the press that there have been dozens or hundreds of amyloid lowering trials, people often exaggerate this, none of them has worked, should not be believed. That's not scientifically rational. These were trials that failed for a variety of reasons and did not actually prove lowering of amyloid. The first real proof of lowering of amyloid in a clinical trial was with the PRIME study at the beginning of the aducanumab development program. And that was quite a striking observation, regardless of what one has finally concluded about aducanumab's utility, and certainly its flawed development process, no question about that. It lowered amyloid in a dose-dependent fashion by amyloid PET, and that predicted in this small trial some degree of cognitive benefit. We also know that lecanemab, which is perhaps the antibody we should talk the most about, is now looking very attractive. Chris and his colleagues published beautiful data from this trial in the New England Journal of Medicine. In our own lab, we showed lecanemab beautifully immunodecorates isolated amyloid fibrils that represent part of the oligomeric A-beta that I've been talking about earlier. And of course, they decorate skeins of amyloid in the brain. So lecanemab likes to bind avidly to amyloid fibrils, and we know what lecanemab has done in the clinic. In particular, it met its primary endpoint and all four of its secondary endpoints with high statistical significance. There was a separation of drug versus placebo that began at 6 months with apparent widening through 18 months. I'll show you that. Safety is an important consideration, and ARIA occurred in about 12% or so of patients from all genotypes, but only 2.8% was symptomatic. That's another issue that we need to be very careful about. The FDA granted that accelerated approval in January, and there's an amendment for full approval. And I want to mention that the whole thesis of this debate is, will we generally accept amyloid lowering as almost the sole standard for approval? I don't believe that for a moment. It depends on the quality of the data, the quantity of the data, and how good the biomarker evidence and clinical evidence is. But for accelerated approval, it may be imperfect clinical evidence, as Holly made clear in the FDA standards. And for a validated biomarker accompanying that imperfect clinical evidence, I think amyloid plaques are almost or virtually validated biomarker now. And 95% of these phase 3 lecanemab patients went on to open label extension, now getting it largely sub-Q. We know that in these trials, these 3 antibodies lowered tau. I won't go through the details of this, but if you want to lower tau in people with Alzheimer's disease, amyloid lowering is frankly, the only proven way so far to get tau lowering rather than approaching tau directly. Now, this bit of evidence from the lecanemab Clarity AD study shows the very substantial amyloid lowering, which I'm believing is a surrogate marker of clinical benefit over time. And so that spread over 18 months. It began at 3 months, and it was very substantial, 59 centiloids in 18 months versus placebo. And as one cognitive output, the ADCS, MCI, ADL, one behavioral output, I should say, there was spreading again over the 18 months. And this again achieved considerable significance in terms of clinical meaningfulness. This was about a 37% slowing of decline in ADL. And that's what my patients tell me they're most anxious to do, is to not have the sense that they're losing ground as much as they might otherwise. So I think clinical meaningfulness is being achieved. At the end of the day, clinical meaningfulness may be the answer to the question, if I offer you this agent with all the information we have about it, would you prefer not to take it or take it? And many patients are now coming forward because they believe the Clarity AD trial was so well executed and the antibody is of interest that they will listen to a discussion of that. Notice here in the middle that all of the 6 components of the CDR sum box, all the domains, showed benefit individually. So this was something not isolated to one test. In an earlier study, the amount of hippocampal volume and the amount of FDG PET signal was also correlated with amyloid positive individuals, shown here, versus amyloid negative of the same age. This was from the Harvard Aging Brain Study, Risa Sperling's preclinical study. And in that same population, in people who had neither A-beta nor neurodegenerative signs, there was no loss of function. But here in the dotted line, if they had A-beta buildup and had some evidence of neurodegeneration, then over 3 years, there was a striking decline. And these, again, are in people who are still normal at entry into study. Amyloid and tau PET positive, cognitively unimpaired individuals are at high risk for future cognitive decline. That comes to the heart of the matter as to whether we should accept amyloid accumulation as a worthy endpoint in itself for predicting less cognitive decline in the future. And this study shows it. We saw, for example, that 14 to 19 times higher.
Yeah, we're at time, Dennis.
Okay.
Yep.
And then I think I'm going to just show this last slide. And that is that the overall analysis by FDA shows that some trials in the beginning didn't lower amyloid and therefore didn't help cognitive function. And a number of trials here, including the Clarity AD trial, which is shown in the circle, did achieve both of those points. So
all right, thank you. Brilliant. Next up, does Dr. Selkoe on screen shares. Dr. Van Dyck will put up his slides. Dennis, feel free to mute up and whatnot. Chris will be presenting for 20 minutes. Then we'll open it up for Chris, Dr. Van Dyck, and Dr. Selkoe to 5 minutes of conversation. And then the vote will go up to vote on the question of amyloid PET as a suitable surrogate endpoint for clinically meaningful outcomes in the accelerated approval of Alzheimer's disease drugs. Chris, take it away, 20 minutes.
Sure. Well, thank you, Jason. You can see this and hear me okay?
Yep, you're good to go.
Okay. So in any case, obviously, as my title says, I'm going to argue that amyloid PET is not yet a suitable surrogate endpoint for clinically meaningful outcomes and the accelerated approval of Alzheimer's drugs. Now, if anybody is expecting me to bash the amyloid hypothesis or bash, biomarkers as as worthy surrogates in general, they're going to be sorely disappointed. I'm really going to argue this quite literally. I'm a fan of all of Dennis's work that he has shown. I believe the amyloid hypothesis, although as one of probably multiple hypotheses of Alzheimer's disease. Here's my disclosure, which Jason already went over. And again, I have no personal financial stake in the outcome of any of these drugs. So Holly gave us, a wonderful introduction to accelerated approval from a regulatory standpoint. And what I really, want to say here is, just because it's legal doesn't mean it's always a good idea. It doesn't mean we need to apply it, in our situation. She already described the development of the accelerated approval pathway in the context of HIV/AIDS drugs. And I do think it's important to really note that Alzheimer's currently is a very, very different situation. Our current therapeutics, as much as they may have meaningful benefits, they don't immediately save lives, offer a cure, and offer people just fully normal lives, ever after. They also require enormous infrastructure of, biomarker evaluation, drug administration, and safety monitoring programs. We can't just go on and off with this program, based on further evidence. We really want to be more sure. And I would argue that in this setting, the standard of reasonably likely, as Holly referred to, really should be different for our current Alzheimer's therapeutics and should probably approach the higher standard of surrogate biomarker full validation. You'll also see that I'm inspired by the landmark paper by Fleming and Demetz back in 1996, surrogate endpoints in clinical trials, are we being misled? And I'm going to refer to some of their tenets. The second point here is that an effective surrogate endpoint must reliably predict the overall effect on the clinical outcome, not just part of it. And in practice, this requirement frequently fails. Another point is that therapeutic effect on clinical endpoint may be underestimated if there's considerable error and noise in the surrogate endpoint. Fleming and Demetz are really, believers that surrogate endpoints are useful in phase 2 proof of concept trials, but in definitive phase 3 trials, except for rare circumstances, they tend to believe that the primary endpoint should usually be the true clinical outcome. This is an outline of the points I'm going to cover. I'm going to argue, one, that amyloid PET only measures fibrillary beta, which may not capture the full therapeutic effect on the clinical outcome. Number two, standardized measurement of amyloid PET remains unvalidated and may introduce significant error and noise. And finally, three, the analysis of the actual trial data from amyloid-lowering therapies has not yet established a threshold of surrogate efficacy. So starting with number one, I'm always uncomfortable when I hear my colleagues talking about how these antibodies, these amyloid-lowering therapies, how they clear amyloid, as though amyloid is amyloid, a unitary thing. So I think it's really important to talk about the different conformations of amyloid. And Dennis has, not surprisingly, he's already done my work for me. So all I'm going to point out in this slide is that, and again, Dennis is really the leading expert in the world in informing us that it's these soluble aggregated species, oligomers, protofibrils, that appear to be the most neurotoxic and probably most critical for their impact. And when we further talk about the antibodies, I think it's important to talk about, the different epitopes and the conformations of A beta that they target. So I'm going to be focused on the 4 in red. First of all, with gantenerumab, lecanemab, and aducanumab. By the way, I'm focusing on these in red because these are, the plaque-clearing antibodies that are in that have been in late-stage development. But Gant, lecanemab, and aducanumab, are all what we would call N-terminal antibodies, and the N-terminus of the A-beta peptide is still visible in aggregated species, including oligomers and fibrils. Conversely, donanemab, binds a pyroglutamate form of the peptide that's associated with plaques, and donanemab appears not to target any of the soluble species. Well, that would, despite all of these different conformations of A-beta, our key surrogate endpoint that we're talking about here, which is amyloid PET, only measures one of them. It only measures fibrillar amyloid and plaques. Everything else I would call, the dark matter on the PET scan. We're just not assessing it, and it is probably extremely important. So this slide, is showing an antemortem postmortem case, a PIB PET scan in the third panel acquired 10 months before death, and then an autopsy analysis looking at plaque density using a fluorescent PIB derivative. And you can see there's a very close correspondence there. But again, amyloid PET is only targeting one form of amyloid. And this might be okay, if the antibodies in question targeted soluble versus fibrillar species equally. In that case, the plaque on PET could be kind of a proxy for the overall picture. But that just isn't true, at least based on the preclinical data that we have so far. So lecanemab, is actually quite relatively selective for soluble protofibrils versus fibrils, whereas gantenerumab and aducanumab, target both fibrils and oligomers, a little bit more selective for fibrils in each case, actually. And donanemab, as we've seen, is fully selective for the fibrillar species. So these antibodies differentially target the dark matter of soluble aggregated species, and they're not equally represented, by their PET data. So point number 2 is that standardized measurement of amyloid PET remains unvalidated, even with the development of the centiloid scale. And I find many of my colleagues, who are kind of users of the centiloid scale, really treat it, as this sort of divine thing. I mean, it's like, at least like the standard meter, at the Palais de Luxembourg, what could go wrong? Whereas the people who actually develop the centiloid scale will tell you it's a lot more like, making sausage. And there's a lot, there's a lot of, crude work that's been done that's not fully validated, and they really emphasize the need to work further, on the centiloid scale. Now, again, the centiloid scale, was introduced in order to provide standardized and quantitative amyloid plaque estimation by PET. It's anchored on a 0 to 100. It's anchored on 2 data points, 0 being the average value in young controls and 100 the average value in mild to moderate Alzheimer's disease patients. So conversion between tracers, is complex. It's based on each tracer's comparison back to, the original tracers, carbon 11 PIB, which is not used in trials. So all of these tracers that are used in the trials, the F18 labeled, for example, if you're going to compare FLIR-beta peer data to FLIR-beta Ben data, it's really based on equations that come from two tracer comparisons, FLIR-beta Ben to PIB and FLIR-beta peer to PIB. And the equations are derived from fairly small samples, and they have a certain amount of error, not surprisingly. And there's been no subsequent validation of the overall process. So, I'm pointing to a vulnerability of intertracer comparisons with centiloids. I mean, do we have any evidence that anything, might be wrong here? And I would argue that we probably do. What I'm graphing here, or what we're graphing here, is now the relationship between baseline CDR and baseline centiloids in all of these same trials that we're going to be talking about, or that we are talking about, with the 4 antibodies. And this is not yet looking at PET as a surrogate for the CDR. It's simply asking the question, do the baseline data appear consistent, with regard to the sort of severity level of the different samples? And overall, there's an association, but there's a lot of noise here. So if you look on the upper right corner, you can see that the donanemab data points are in red, and they're near those gantenerumab data in blue. The donanemab centiloid values are about 10 points higher than the gantenerumab data, whereas the patients are of similar severity. And if you look in the lower left part of the graph, now you see the aducanumab data points in orange and yellow compared to lecanemab in the light blue. Here, again, aducanumab about 10 centiloids higher, and yet the aducanumab participants are actually significantly milder than the lecanemab. So this is concerning, and in both cases, note that what we're looking at is that the studies that use florbetapir, which are in the red, yellow, and orange, are showing unexpectedly high values relative to the studies that use primarily florbetaben. And this kind of intertracer issue is already a problem if you're doing a study that uses multiple tracers. But it's a deadly problem if you're trying to compare data across studies, which is exactly what you need to do when you do a meta-analysis to look for, a surrogate, surrogate indicator of efficacy. So apart from tracer differences, next we have scanner differences, which are also probably quite important. And these data just come from an anecdotal case that was described to me by Chris Rowe, one of the world experts in these issues. So he described here his two different scanners of different resolution. The vision is a good millimeter and a half better resolution than the TF64. So when he scanned the same patient on both scanners, he got different centiloid values. It's only 4 centiloids at the level of 0 centiloids, but when you get to 50 centiloid range, it's about 10. And when you get to 100 centiloids, which is around where a lot of these trials start, it's 20 centiloids difference. So I would say that the standard meter maybe needs to be replaced by the king's foot, because what we're really talking about is it depends, who's the king with the king's foot. And that's kind of like, it depends what scanner you're using, and it depends what tracer you're using. And this factor, does, I think, introduce noise that could lead to underestimation of a therapeutic effect on a clinical endpoint. So the last thing, I want to talk about then is the analysis of actual trial data from the amyloid-lowering therapies, and I'll argue that we don't yet have an established threshold of surrogate efficacy. So these data, as we'll see, are examined through meta-analysis of the relationship between the surrogate and the clinical outcome. And we're very, fortunate to have the work of the FDA biostatisticians who worked on these problems that they've published and explained very well, that provide a good roadmap. So even if we don't necessarily agree with their decisions and conclusions, they've really benefited, the field tremendously. And in this slide, I'm going to follow, one of the meta-analyses that was published by Zhu et al. These are FDA biostatisticians, and here they're looking at the relationship between drug placebo differences in % change in centiloids and change in CDR-SB. These are data from aducanumab at all levels of development, and then the donanemab phase 2, but momentarily I'll add in the newer trial data. First of all, they did include the data from the PRIME study, which Dennis referred to. Now, I really have to say these data do not belong in a meta-analysis. They are, these are observed case analyses that were done in small samples. There were very high discontinuation rates in these studies, especially due to ARIA. A lot of the patients didn't come back, they weren't followed. And this results in a substantial attrition bias because people who don't come back tend to be doing worse. And that's why you see this, huge effect, of 1 full point on the CDR-SB, which never occurred, ever again, with aducanumab. So for those reasons, I'm just going to remove those points from the graph. And now I'm also going to add in the new data from lecanemab and gantenerumab. Dennis has already referred to lecanemab, Clarity and the Gantt data are the red boxes. Those are from Graduate 1 and 2, which was a negative study. So now, do we have enough data, to define a threshold? By the way, let me just point out that this point here, the eMERGE study, if you can see my cursor, is actually misplaced. Some of the data looks skewed on this figure. It was not a 0.6 difference on the CDR, it was about 0.39, so it really belongs up here where my cursor is. But in any case, the FDA statisticians acknowledged that based on the data before these points are added, that the threshold really had not been robustly defined, as showed in the quote down in the lower right. So now, do we have enough data for a threshold? Well, if we did, it probably goes where this little dashed line is, right? We kind of cram it in between the eMERGE study, but to the left of the negative Graduate 1 study. So this is just to point out how really dicey this kind of thresholding is at this point. And by the way, this is looking at percent change in centiloids. More common is to look at sort of absolute centiloid change over time, not percent change. And if you do that, by the way, the Graduate 1 point goes way over to the left of these 2 positive studies, and it makes it harder then to even think about, a threshold. And speaking of that, I'm going to now flash up the Graduate 1 and 2 data for gantenerumab. And this, again, is just looking at, amyloid plaque removal in the 2 studies, which, of course, is our topic. And you can see in the title, that it was found to be below expectations. But if we're going to use these data as a surrogate, it doesn't really matter what the expectations were. We've got to just look at the results. And the results are that gantenerumab actually in Graduate 1 actually cleared more plaque, by centiloids than, I'm going to pop up Clarity AD, which Dennis showed you. It cleared more amyloid than did lecanemab. So, this raised a number of questions. One of the questions that came up right away was, well, maybe it's the slow kinetics of amyloid clearance with Graduate 1. You can see that at 1 year, less than half of the plaque clearance had occurred, whereas in Clarity at 12 months, most of the plaque clearance, is present. So that's very interesting. But I would say, if we have to think about kinetics for surrogate biomarkers, then we have not been doing that, and we need to really go back and do some more homework. And I don't think that we're ready to do that at this point. By the way, another explanation, couldn't it simply be that, in lecanemab on the right, which is targeting soluble species much better than gantenerumab, couldn't it just be that it's clearing more of that unmeasured dark matter? And that's why, it achieves efficacy, even though the plaque clearance wasn't as much. Another thing that, that these data, brought up to many people is that it may not be about the amount of plaque you remove. It may be about the amount of plaque left after removal. And that's certainly a reasonable, thought. So, for example, if we look here, we can see that all of the trials that have achieved a final value of less than 25 centiloids have been positive. So maybe there's something to this. I would argue, though, that this is, kind of the flavor of the month, making it up as we go. This is otherwise known as, something that probably is hypothesis generating, and maybe now we need to test it out, in a few trials before we actually use it as a surrogate. This also may be too restrictive a threshold because, again, there may be antibodies that don't, get to this level but are still efficacious by targeting soluble species. A final thought I have is, why are we attempting any of this anyway? A useful surrogate biomarker should accelerate the process with shorter trials and fewer participants. So far, amyloid PET has not shown anything that could be accomplished in shorter trials. It is usable with fewer participants, but I would argue that we still need full samples for safety. Why not continue to acquire the clinical outcomes and analyze them? And this goes to Fleming and Demetz's point that in these trials, except in rare circumstances, the primary endpoint should usually be the true clinical outcome. And so in quick summary, I think I'm probably out of time. So I won't even review this, but there's my summary slide, and I thank you for your attention.
Right. If Dennis could unmute video and audio. Chris, take down your slides, and I'm going to just put up the question for vote, which will happen in about 5 minutes. I'd like to give Chris and Dennis the opportunity to exchange for about 5 minutes. Actually, not about 5 minutes, for strictly 5 minutes. Then the vote will go up. Let me remind everyone that you also can send to that email address, tjcasey@upenn.edu, any of your comments around your vote. It's understood if you do that they'll be part of the public display of this recorded webinar. And also, I want to thank the leadership of the Alzheimer's Clinical Trials Consortium for making this webinar possible, and the members of the Internal Ethics Committee for brainstorming and putting together this project. Chris and Dennis, please, the floor is open for the two of you for 5 minutes. The clock has started.
Well, I certainly think Chris made some very rational arguments about why amyloid lowering as a surrogate marker by PET is not quite ready for prime time. My perspective was that it will likely become that way quite soon. So I think in the next few years, amyloid lowering will be a very clear indicator of likelihood, a reasonable likelihood of clinical benefit. And I think the data I reviewed already show that it correlates with the benefit. I accept the appropriate technical arguments that the precision of amyloid PET is not yet uniform. In terms of cross comparison between studies, I was thinking of a particular antibody that also has safety for sure. That's the key phenomenon, and then is used in a uniform way in a rigorous trial, as I think it was in Clarity AD. And in that example, if there is a uniform PET ligand that is done relatively uniform, and we need to see the details, Chris would know them, of how much variation there was across sites and in PET scanning. My prediction would be that that will correlate quite well with cognitive decline, and I think it already is. Certainly the biology predicts that over time, amyloid lowering will be meaningful for clinical outcome because the small oligomers that Chris brings up as a problem potentially are in tight equilibrium with the plaques. In fact, the plaques are bundles of oligomers that come on and off. And there's some beautiful scientific evidence to suggest that the penumbra around plaques is an oligomer-rich area where there's destruction. So a lot of this depends on one's perspective on our sense of urgency. I agree that we have no ideal surrogate marker today, but I think that amyloid PET lowering is quite strongly associated in different studies with the clinical benefit. And I do think, as Chris says, it depends on the degree of lowering. So it may not be there yet in terms of the technical performance of PET scanning with the ligands we use now and the scanners we use. But biologically, it's very likely that if you lower amyloid plaques by PET, you will see an effect on oligomer species, which I agree are particularly bioactive. And that should be able to decrease patient numbers in trials, as will tau plasma biomarkers, and make it more likely that amyloid reduction will lead to benefit.
Chris, you have 2 minutes and a half, roughly.
Well, so Dennis, I appreciate that. I actually felt in watching your presentation that there was really very little I disagreed with. I thought, we were perhaps arguing two different parts of the question. I think you were really providing all of the great evidence in favor of the amyloid hypothesis, but then when you get down to the brass tacks of really, can you use amyloid PET as a surrogate? I'm not sure that all of that changes, the answer to the question. One thing, when you refer to, let's say, oligomers, as being in tight equilibrium, monomers, oligomers in tight equilibrium with fibrils, that may be the case. I don't know if that's fully the case, in vivo, but if it were the case, you still care about more than one of those species, and PET isn't measuring them. And again, if the antibodies differentially target those species, then PET doesn't equally represent, all of the antibodies. And I think that really gets to the whole issue, that Fleming and Demetz, raise, which is that if you're going to use a biomarker, it's got to fully, reflect your true clinical outcome, not just part of it.
All right. Well,
I think one point about that is that we will couple this with tau outcomes. So that'll be very much more instructive, amyloid lowering and tau effects. That may give us a much more compelling set of surrogate markers that may predict efficacy.
Well then, thank you, gentlemen. And again, I want to thank the leadership of the Alzheimer's Clinical Trials Consortium for making this possible, and the members of the Internal Ethics Committee for working to put this together, and also Taryn Casey, head of the communications team for the Penn Memory Center and the Alzheimer's Disease Research Center. Now we'll put up the vote on the question, which is before you on the screen. You see the vote here. Please vote yes or no. You'll also see Terry Casey's email address is your opportunity to send your comments on your vote. If you do send comments, we assume that you want them, they will be published. We'll compile them together. This presentation is being video recorded for presentation on the ACTC website, as well as the Penn Memory Center Alzheimer's Disease Research Center website. We will let you know when that all goes up, so it can be viewed. And it looks like we've maxed out the voting at 75% of the participants. A total of 92 individuals have voted, and with 100% answered, we have to the question, amyloid PET is a suitable surrogate endpoint for clinically meaningful outcomes and the accelerated approval of Alzheimer's drugs. The votes for yes are 17 out of 69, and the votes for no are 52 out of 69. I will let all just simply take the vote and ponder it as they see. Again, please send your comments about your vote to the email there on the screen, tjcasey@upenn.edu. This is being recorded for subsequent viewing, and comments that will accompany that you send, and we'll let all participants know when that's all available. I want to thank Drs. Van Dyck, Selkoe, and Fernandez Lynch for their fantastic presentations, and I want to thank you all for attending. Again, science proceeds by open controversy and debate and discussion. I think we've achieved that today, and I look forward to meeting you all again for another discussion of some of these contentious issues in ethics and science. Take care, everyone.
Thank you.
Thank you.
Okay.